Hi, this is Arjit Kara and I work with the Lee Lab in the Department of Biology. I am working on a project that aims to characterize genetic mechanisms involved in measuring day length, also called photoperism, in Drosophila melanogaster. Many organisms are known to respond to seasonal day length changes. This phenomena is called photoperism. Photoperism is also known to regulate seasonal behavior and physiological processes in many organisms. Photoperism can impact human health, and in the bottom right you can see during the spring and the summer months when we have longer days, there is a rise in manic episodes. And then during the fall and winter months when the days are shorter, there is a rise in depressive episodes. There are many reports supporting the view that an organism can measure the day length through an endogenous 24-hour cellular clock, which is termed the circadian clock. The circadian clock is known to regulate many biological and metabolic processes. However, little is known about the genetic and molecular mechanisms of how the circadian clock is involved in measuring seasonal or day length changes. So here we have hypothesized possible interactions between the circadian clock and photoperism. First is that circadian clock and photoperism do not interact at all. The second possibility is that either the circadian clock controls the regulation of photoperism or photoperism regulate the responses of circadian clock. And the final possibility and the most likely possibility is that there are some overlap in mechanisms. The goal of this project is to better understand the genetic mechanism of photoperism and the role of circadian clock in photoperism using Drosophila melanogaster. In the current study, we performed a genome-wide association study on photoperism using Drosophila Genetic Reference Panel, which consists of a fully sequenced inbred line created from a natural population. We assessed the photoperiod response of the flies by using an assay called the Tilcom recovery time. The flies were placed under two different photoperiod conditions, short day and long day. Then the chill coma was induced by placing them in 4 degrees Celsius for 60 minutes. Then the time it takes the flies to recover from this coma was measured. We performed this assay for all the 205 DGRP lines and then performed the GWAS on this data. The GWAS in the DGRP population under different photoperiod conditions resulted in 40 candidate genes responsible for photoperiodic regulations. We further analyzed the knockout mutants of top 4 candidate genes for their photoperiod response and circadian period. PKC delta and GLC-ATP mutants showed significantly different photoperiod response from that of the DGRP208 wild-type strain. Since in the GOS, PKC delta had 2 independent SNP variants associated with the photoperiod phenotype, and here the PKC delta mutant has significantly different photoperiod response from the wild-type, we chose to analyze this gene in depth. Here you can see PKC delta has 3 transcripts, and it is located in the X chromosome. It is also known to be involved in signaling and signal transduction pathways. We also looked at the 2 different mutant alleles of PKC delta, the piggyback mutant and MIC mutant, and observed their photoperiod response compared to the DGRP wild type. We observed that they had different photoperiod response from one another and compared to wild type. In order to test if these photoperiodic candidate genes would be involved in circadian clock, we performed the affiliate phenotype assay, and out of the 4 candidate genes, GLC-ATP knockout mutant had an altered circadian period phenotype. We then analyzed the non-circadian clock mutants and their photoperiod phenotype, concluding that clock mutants have a photoperiod-specific effect. This suggests that there are some shared regulations between the clock and photoperism in Drosophila. The current study rejects the hypothesized model about circadian clock and photoperism being separate entities with no shared regulations. Taking this data together, we identified molecular components of photoperiodic response, which led us to new insights on how circadian clock is involved in insect seasonal response to photoperiodic changes. So far, we have only analyzed 1 candidate photoperiod gene in depth. However, the GWAS produced 40 candidate genes that could play a role in photoperism. This study overall sheds light on potential genes that could be further studied in order to characterize complex mechanisms regulating photoperism.